Our next segment is a great example of convergence of disciplines. It's my pleasure to introduce Professor Ariel Furst. Professor Furst joined MIT's Department of Chemical Engineering in August 2019 and is today the Paul M. Cook Career Development Professor. Her lab combines electrochemical methods with biomolecular and materials engineering to address challenges in human health and clean energy. So what you're about to see and hear is the convergence of biology and chemical engineering in action. That's very exciting. Professor Furst.
Thank you. Let's see if I can get my slides up. Oh. So today I'm going to talk about two different projects. I know my title was only about CO2 upcycling, but I really wanted to address the question that was asked to the last speaker, which is, can we both remediate the damage that's already been done while also improving our technologies so that we don't continue to do more damage? So my lab focuses on technologies that improve global equity. We like to think of current technologies as divisive. They serve the 1% for the most part. So our goal is to make technologies that can be applied broadly and that everyone has access to do. And so today I'm going to focus on two projects. One, to turn CO2 into valuable products, and the other, to overall decrease the amount of CO2 we're producing. And we do this using two different core technologies that I'll focus on. The first is microbial coatings. You can think of them as tiny little suits of armor for microbes. And the second is DNA Velcro, which is using the materials properties of DNA rather than the genetic code. So this first project I'm going to talk about was done by my postdoc Gong, and someone who started in my lab as a freshman during the pandemic, Pris. She's now a senior. So the question we were asking is, what are some main contributors to CO2 production? And one of the key processes is the Haber-Bosch process. That's how chemical fertilizers are made, and it's allowed us to feed the current global population. The problem is, this produces a significant amount of greenhouse gases. It's approximately 1 billion miles driven in gas engine cars each year. And furthermore, nearly 20% of what we apply is wasted as runoff. And this doesn't even include the skyrocketing prices that are preventing farmers from actually using the correct amount of fertilizer. So we take inspiration from nature. How does nature fertilize the soil? Well, it uses a consortium or a group of microbes, things like photosynthetic organisms that can convert light into energy, decomposers that can circulate material from dead matter, nitrogen fixers, which is what we'll focus on today, that actually make fertilizer in the soil at the site of plant growth. And then every one of us actually has more microbes in and on our bodies than mammalian cells. So we are mostly microbes. And we like to think of microbes as tiny little chemical factories that have, through evolution, optimized the production of whatever we're trying to produce. So as chemical engineers, the hard work is already done for us. So when we think about microbial fertilizers, we can think about improving the quality of the soil, improving crop stress tolerance, and improving growth and yield. The problem that people have been facing is that in order for these microbes to work, we have to get them from the factory where they're produced to the fields. And so we've long known how to grow microbes and study them in the lab. We can even grow them at large scale in fermenters. The problem is many of the microbes of interest for soil are very delicate. So we can't actually formulate them or transport them. Well, my group has developed nanoscale coatings that self-assemble on microbes like a little tiny suit of armor. And this has allowed us to formulate and transport any strain we want. And so today I'm going to talk about 3 different examples B theta, which is part of our native gut microbiome, and is thought to be a treatment for colorectal cancer. It's worked great in mouse models. The problem is we can't actually produce it for human consumption. The second is B subtilis. Many of you might take the spore of this as a probiotic. The problem is the spores are like tiny little protective coatings that keep those cells as spores, so there's really no beneficial health effects. And finally, we're going to talk about P. chloraphis, which is a nitrogen fixer. It's very temperature sensitive and highly unstable. But if we could deliver this to plants, we hypothesized that it would help plant growth and production. So we coat microbes on what's called metal phenolic networks. They form in seconds, which you'll see here. That blue color is the coating forming on the microbes. They're inexpensive, safe, and easy to form, and we can actually tune their properties to protect against different stressors. They're comprised of polyphenols. These are things like tannins that are found in coffee and wine, or green tea extract, as well as simple metal ions that you take in daily supplements, like iron. And they assemble into these complex matrices that form on anything in solution, including the microbes. We can see here they're also really easy to see and visualize. So all these different colors are different metal phenolic networks forming on microbes. And these colors form because of ligand to metal charge transfer, which is basically just the energy of those bonds forming. But it's a really easy visual way for us to tell that we've been successful. We can also look at cells coated in these and see that there's a very uniform coating. So this technique is called TEM, and basically it looks for metal ions. So normally you have to stain cells to get resolution. On the left here, you see an unstained cell that's not coated. It's very fuzzy. On the right, we can actually use the metal ions in our coatings as the imaging agent. So we can see this very consistent coating with very high resolution. And our coatings form this really unique core cell structure. So we can stain the coatings with a fluorescent molecule. So we have uncoated cells, we obviously don't see anything. But on the coated cells, what you'll see is darker red around the edge and lighter in the middle, indicating that they're actually forming all the way around the cell. These coatings don't affect initial cell viability, which is really important, right? We don't want to kill the cells before we use them. So this is a very simple live dead cell stain. Green means they're alive in the left, red means they're dead in the middle, and then we have them overlaid. And you can see that for both uncoated cells on the top and coated cells on the bottom, we have the same number of living cells. So what can we do with these coatings? Well, so far we've shown protection for lyophilization, oxygen exposure, heat, and humidity. And so we'll go through examples of these one by one. So lyophilization or freeze drying is a core component of the majority of formulation for any sort of microbial product. And we can see here, we just have a live cell stain, that the uncoated cells, the majority of them are dead. In contrast, the coated cells are surviving with much higher viability. We can also look at an anaerobe. So B theta, the microbe that I talked about that can be used to treat colorectal cancer, is an anaerobe, so it doesn't like oxygen. Well, we can see that for the uncoated cells, if we freeze dry them and store them under oxygen, they all die. In contrast, our coated cells not only survive, but they grow to a normal steady state within a normal amount of time. So we really haven't impacted their growth or proliferation. Importantly, I showed you a bunch of different components we could use for these coatings. So what these two graphs are is just different combinations of those polyphenols and metal ions. And what I want you to take away from this is that not all the coatings work effectively for every stressor. So that's where the optimization here comes in. We can see this when we heat cells. So these are P. chloraphis, those nitrogen fixers. And we can see that based on the polyphenol we use and the metal ion, we get very different viability of cells, and that just means the number of cells grown. So on the left, that's the peak of that curve, and on the right, that's the tall part of the bar graph. But when we optimize this, we get really incredible results. So on the left here, we can see cell viability as a function of temperature. For our uncoated cells, anything above refrigeration kills them. For our coated cells, we can show that they survive with nearly equivalent viability up to 30 degrees Celsius. But more importantly, we can stick these in an oven at 50 degrees Celsius, which is 122 degrees Fahrenheit, for 5 days, and we have cells survive. That's a temperature that can cook an egg. And so we wanted to see, maybe, there we go, if these actually helped plant growth. So we looked at alfalfa sprouts and bok choy, and we looked at no cells added, just freeze-dried cells, fresh cells, and our coated cells that had been stored at 50 degrees for 5 days. And what you see here is a clear impact of our coated cells. We can see statistically significant improvement in growth for both of these crops. And we can see here that when we compare them side by side for 10 days, we get very similar efficiencies, even though they're very different crops. So, what we've been able to do so far is to improve microbial manufacture to the point where we can actually produce microbes that survive freeze drying, which is the first key step in terms of formulation. We can then protect anaerobes from oxygen stress, and we can protect temperature-sensitive microbes from both high heat and humidity, which are key stressors that normally prevent the use of microbes, especially in agricultural situations. And so as we move forward, we are continuing to try to optimize this, and our next step is actually field trials to apply these to cash crops like corn and soybeans. But we overall anticipate that this will be a significantly equalizing technology by decreasing the price of fertilizers for those who use them. Okay, so second half of the story. So now we figured out a way to decrease CO2 emissions. We don't have to use hover bosch as much. We can use these microbial fertilizers. But there's already a lot of CO2 in our atmosphere, and in order to mitigate climate change, we have to remove some of that. Well, what if we could not only remove it, but also make it profitable? So this work was also done by my postdoc Gong, as well as a postdoc Thomas Gill, and an undergrad, Rachel, who started in my lab as a sophomore. And when we think about carbon dioxide, we can think about adding different numbers of electrons to it to get different products. That's fundamentally all we are doing when we're reacting carbon dioxide. And what I have here is the commercial value of each of these products. And you can see there tends to be a trend that the more electrons you have to add, the more expensive the product is. And that's because adding electrons is difficult. So we wanted to see if we could reduce the amount of energy that it took to do these conversions. One way we can think about doing this is with electrochemical conversion. I'm sure many of you have heard of electrolyzers. And the goal of these is to make a completely circular carbon economy. If you take renewable energy to power your transformations and carbon dioxide and water, we can use electrochemistry to turn that CO2 into products like ethylene or ethanol that can then be recycled into chemicals and fuels. And then you can think about just starting that process over. The problem is that the amount of energy we need to put into these reactions is very similar to many other reactions that happen in water. Things like hydrogen evolution, oxygen reduction, water splitting, which are great in some cases, but are off targets for us, and those lower the efficiency of our reactions. So we again looked to nature. Nature has made catalysts with unparalleled sensitivity and specificity as well as activity. These are called enzymes, and they run pretty much every biological process in our bodies. And specifically, we focused on enzymes with a core active site called hemin. It's got a structure called a porphyrin, with a metal center in the middle. And there have been many catalysts made that actually mimic this structure, which you can see below. And so we started by just looking at catalysis with these small molecules. And so what you'll see here in each of these is just the amount of current generated, which indicates how much we're transforming whatever's in the system, in either nitrogen in the black or in the color in the second graphs. And so if you look to the left of each of the graphs, you'll see that the peak is significantly lower for the colored one. That indicates catalysis is occurring, which is fantastic. The problem is these catalysts degrade nearly immediately. So within seconds, we see this current change, which indicates those catalysts plating onto the electrode and ruining both the catalyst and the electrode. So although these catalysts have been used for years to show small-scale transformations, they really haven't been industrially viable. So we wanted to think about whether we could actually use the inherent materials properties of DNA to improve this catalysis. So why DNA? I'm sure many of you think of DNA simply as genetic code. Everyone learned the central dogma of biology in high school, right? Well, if we look at the three-dimensional structure of DNA, there's a whole lot more there than just the genetic code. It's got this very specific structure formed by these two individual strands. And importantly, those strands are specific to one another, and they recognize each other based on sequence. So we can actually think of DNA as like a tiny little piece of molecular Velcro. Velcro adheres because of electrostatic interactions. DNA adheres because of hydrogen bonding. It's a reversible interaction, but it remains very tightly bound under the proper conditions. And if we even look at those porphyrin-based catalysts in a native protein structure on the left compared to a DNA strand on the right, we can see that modeled here, it actually looks like that secondary sphere, so the area one step outside of that catalyst, is very similar. So we thought that using this DNA would not only allow us to specifically place these catalysts, but also improve their local environment. And so what I'm going to show you now is some data where we were able to modify one strand of DNA with the catalyst. We can modify an electrode with the complement. And when we mix those two solutions, that catalyst naturally hybridizes to our surface with about 98% efficiency. And what we can see here is when we just add the DNA, so this isn't even on an electrode yet, we see we significantly improve the stability of these catalysts. So on the left, we see the free catalysts decrease in current, crapping out on our electrode. On the right, we see we can continually increase the potential we're using. So more and more power that would normally kill that catalyst, and it doesn't destroy the catalyst at all. So we've saved one of the core problems of these catalysts, their instability, by just adding the DNA in the first place. We can then look at these selectivities in different solutions. So what we have here is just 3 different potentials, and we're looking at selectivity for hydrogen versus carbon monoxide. So carbon monoxide is syngas, industrial important product. And we can see that we get significantly more CO produced with that DNA. So that means we're actually shifting the catalytic properties. And even more importantly, if you look on the far left at minus 1.15 volts, that's about 300 millivolts less energy required to do that transformation. So what that means is to get CO from CO2, we've decreased the amount of energy you need by 30%. So that's going to cost a company 30% less to actually use the CO2. And importantly, on the bottom here, what we see is with our optimized system, we get nearly 80% Faradaic efficiency. So that means for every electron we're putting in, we get 0.8 molecules of CO. That is unprecedented. The majority of these porphyrin catalysts in solution look like the rest of this graph. You either get significant amounts of hydrogen production, which is not what we want, or you get nearly no conversion at all. with really low Faradaic efficiency. So being able to get 80% conversion by just simply sticking this to an electrode using a really easy molecule to use is really unprecedented. I should also note that you can chemically synthesize DNA for about 1 cent per base right now. And we're using picomoles per centimeter squared of catalyst versus micromolar to millimolar in solution. So we're adding negligible costs while also decreasing the amount of catalyst we need by 10,000-fold. And so what we can see overall is that DNA allows us to not only significantly improve our stability, but also the amount of energy and the efficiency of how much energy it takes to convert carbon dioxide to carbon monoxide. I also want to note that this means that electrodes for electrolyzers for the first time would be recyclable, because now we're not waiting for the catalyst to plate onto the electrode and to throw out our whole system. Now, we can take our catalysts when they're dead on our surface, just add hot water that unsticks our Velcro and dehybridizes our DNA, and allows us to regenerate our electrode. So we've not only decreased the amount of energy you need, increased the production efficiency, but we've also enabled reuse of electrodes that couldn't previously be reused. And so I've told you about 2 technologies from our lab today that I hope just give you a flavor of the type of work we like to do, which is looking at biology for inspiration, because we think billions of years of evolution have us beat as engineers, and to harness that into really useful and inexpensive solutions. And so I just wanted to thank my group. I have an exceptional group. We've started in 2019, so in the middle of the pandemic, and as you can see, we have a fantastic group of undergrads and even high schoolers. So thank you very much, and I'm happy to take questions now.
Thank you, Ariel. We have some great questions that came up on the Pigeonhole. So the first one is, can your method be applied to other areas, like construction materials, for example?
I'm assuming...
The coating.
The coating, thank you. So yes, it can. These cells will really survive any stressor. So there's been a lot of work in construction materials to make self-healing materials using microbes. We can make that process better and more efficient and more long-lasting by incorporating our coatings that add a 0.01% increase in incremental cost.
Great. The second question I'm really dying to ask, how do you think of all these things? So for example, the armoring of microbes, how did you come up with this?
So we had thought about these materials because they're antioxidants. So many of you probably have seen or have even taken green tea extract pills. That's what we use as a source of the EGCG for coating. So we were really just looking at things that we thought would combat stressors to microbes. And when we added these to our buffer, we saw this weird color change, and then we had to characterize it. So we think it's a combination of the rigidity of these coatings, because they're much more rigid than things like polymers, as well as their ability to scrub reactive oxygen species and also absorb UV light.
Great, thank you. Still on the coating of microbes, what about the bad microbes? Could that possibly produce unintentional results in the soil?
So that's a great question. We protect these microbes in batches, so in a fermenter that has been verified to only have the microbes we want. So it could protect bad microbes, but we're specifically delivering good microbes. And the strains we're looking at are ones that are already found in the soil. So we're just kind of building up what's already there and what's already known to be good.
Very specific question. Can your coatings be used to coat probiotics to suspend them in liquids?
That is a great question, and yes, they can. So the B subtilis, as well as the B theta, are both effectively probiotics. We call them living biotherapeutics because they would be prescription. That's really the only difference in terminology. But we've tried this on 20 to 30 strains at this point, and it works on all of them.
What yearly volume of the CO2 transformation can be reached with your technology?
That is a great question, and that's what we're trying to figure out now. So in the lab, we've used lab-scale electrolyzers, but we're really trying to find companies to partner with who have larger-scale electrolyzers to see how well this scales. Based on moving to from a standard lab scale to kind of this pilot scale, we don't see a drop in efficiency. Our goal is also to use this with seawater, so to remove carbon from a standard carbon sink globally. I hope that answered the question.
The other one, did your experiments for CO2 conversion to other products than CO, can you tune selectivity to a single product or mainly reduce HER?
That is a great question. So I didn't include the data here, but the CO experiments for syngas were proof of concept. Since then, we've been able to specifically tune for methane and ethylene production.
Could metal nanophenolic frameworks be used as corrosion protective coating on metals? Can you tune and play with final colors, produce inks?
Yes to all of the above. So they're mainly thought of as protective coatings as antibiotic coatings because it's known that microbes don't like to stick to them, although we can stick the coatings to microbes. And we can also play with the final colors. So we have really tight control. This field has been studied for about 50 years of this ligand to metal charge transfer. So people have actually used these as hair dyes, as temporary hair dyes. So you have really tight control over the color you end up with.
Great. Can you speak a little bit to the efficiency compared to other technologies?
So it's a combination for us of the decrease in the energy needed. So we only need to put in minus 1.15 volts instead of minus 1.5. So that's significantly decreasing the energy input. We also increase the Faradaic efficiency. So for every electron we're putting in, we're getting more CO out. So overall, we think at about a 1 to 2-fold higher efficiency than current technologies.
Great, and we have one last question. Can you explain further how the CO2 to CO could then be converted to CH compounds?
Yeah, that's a great question. So basically, as you continue adding electrons in water, it just keeps reducing and you end up blowing off the oxygen and either bonding it to another carbon or just continuing to reduce to methane using your hydrogen from water as a proton source.
Great. Ariel, thank you very much.
Thank you.